All right, this is section 2.3 of our biology unit in Science 30. This is on DNA. So far we've looked at sort of what inheritance is, what genes are, and how we inherit certain things using the Punnett square. And next we are going to talk about DNA specifically, so sort of the makeup of it and how it works. So first of all, it says the structure of DNA, which is DNA stands for deoxyribonucleic acid. That's why we say DNA, aka DNA, because that's a pretty big word to say. So we normally just say it's DNA. DNA is composed of chemical units called nucleotides, and each nucleotide contains a phosphate, a sugar, and a nitrogen base. So if we look at, this is like a model of what this piece of DNA might look like. We've got our nitrogen base right here. We've got our phosphate right here, and our sugar right there. Okay, so all three of those make up a section of DNA. So the phosphate is that small little piece. The sugar is, there's lots of different types of sugar. So this is a deoxyribose. That's where we get deoxyribonucleic, because there's they're called nucleotides. But yeah, deoxyribose is the type of sugar. And then the nitrogen base is, I'm going to say the most important part, but it is important because we are going to focus on that part of it, just because they could be different. So there's 4 different types of nitrogen bases you can have. Adenine, which we will signify as A, thymine, which is T, guanine, which is G, and cytosine, which is C. So a phosphate and a sugar part of separate nucleotides bond to each other together, or bond to one another to form the backbone or the sides of the ladder. So when we look at DNA, DNA is actually just literally like a ladder that is twisted. So one side of the ladder is all of this. And it's going to connect to another side that looks almost identical. The only thing that's going to be different about it is the nitrogen bases. Okay, so these are the 4 nitrogen bases that we can have, and the other side of the ladder will connect to that. Okay, so that's that one side. And each of these little individual pieces that have the nitrogen base, the sugar, and the phosphate, that is known as a nucleotide. Okay? Perfect. Nitrogen bases pair up to form the other side of the DNA molecule. So you may have noticed when we looked over here that, let's use this part, looks like it connects into here. So those puzzle pieces sort of fit together, and then this part fits into here. So that's actually written that way because it's true. Whenever we have an A, an adenine, the other side has to be thymine. And then whenever we have a C, we know the complementary part, the part that's connecting to it, is going to be guanine. Okay? And that's simply just because that's what fits together. They fit together like puzzle pieces. So yeah, this is what this slide says, that nitrogen bases pair up to form the other side of the DNA molecule, the other side of that ladder. And A's will always bond to T's, and C's will always bond to G's. Okay? So this is what a ladder of DNA would look like. They call it a double helix because there's 2 sides, double. And then helix means it's in a spiral. So this is something that you will have to remember. You just have to remember that A's are always connected to T's and G's are always connected to C's. Okay? If we were in class, we would do a lab on this, but we are not, so we cannot do that lab. DNA from one human cell would stretch out to be over 2 meters long. How can this all fit into each cell? So just your DNA in one human cell would be 2 meters long. How can it fit in such a small area? Well, that twisted double helix shape makes it compact. So like if you had a straight line, you wouldn't be able to fit as much in. If you put it into a spring, you're going to be able to wind up a lot more in there, and that's what DNA is. It's like really wound really tightly together so it can hold as much information in a really small space. Excuse me. DNA will wrap around proteins, protein spools that are called histones. Okay? So these are what the DNA will wrap around, histones. And all of that will be found inside of your chromosome, okay? So inside of there, we've got really, really tightly wound DNA, which will code for everything that happens in the blueprint of your body, basically. So who discovered the structure of DNA? Well, a couple people found out some things, but really Watson and Crick were the ones that discovered the structure. But it was due to the studies of Chargaff and Franklin, who made them be able to come up with the structure. So it says, Chargaff discovered the 1-to-1 ratio that A's bond to T's and G's bond to C's. That was super important to know. Rosalind Franklin, she took a X-ray picture of DNA and found the image that was left was the shape of an X. So she used an X-ray to figure out what DNA might look like. And then Watson and Crick, they used their information to come up with build a model structure of DNA. And they really wouldn't have been able to figure this out without Franklin's work or Chargaff's. But Franklin, little history here, she actually worked with them. So these guys were like the professors and she was like a student. And she found out all this stuff about the shaping of the chromosomes. And Watson and Crick stole her information and called it as their own so that they could get recognized for, and I think they got the Nobel Prize for figuring out this when Rosalind Franklin didn't get any recognition. So sort of sad, but hopefully something that would not happen today, hopefully. That brings us to practice problems. It says on page 109, you want to do 27 to 32. I'm going to say that we're going to only do 27 to 29. So let's not do quite as many questions, just because we need to continue and get through all of this. All right, so that's the first part of this lesson.